So we are looking at nucleotide excision repair. So the previous video was on base excision repair. Now we're dealing with nucleotide excision repair. Now the difference is very clear. The first is base excision. That is, we are simply removing the base. But in nucleotide excision repair, what we're talking about is that there is an oligonucleotide, that is a sequence, a short sequence of nucleotides that we have to remove. So, of course, base excision repair just deals with removing a base. Nucleotide excision repair deals with a string or a set of nucleotides, which we call an oligonucleotide. So there is actually a distortion or a change, a distortion in the double helix that is recognized by a multi-enzyme complex. So once it is recognized, then the nucleotide excision repair mechanism begins. And how does this start? Now, most of the times from research, the major kind of, or what we have been able to observe so far, the major distortion that occurs is what we call this formation of pyrimidine dimers. Pyrimidine dimers. So these are caused by sunlight. So of course, when, pyrimidine, like I told you, cytosine and thymine are actually pyrimidines. So when thymine and thymine forms a dimer, it could also be when cytosine and thymine forms a dimer. It could be in the reverse order, where cytosine and cytosine form a dimer. This is what we are talking about by pyrimidine dimers. So let's look at it. Let's look at it and let's see how it actually occurs. So I'm going to show you a sequence and we'll look at this together. So let's say we had this sequence. We have G, C, T, A, T, G, C, T, A. Right? What would be at the opposites? Want to try? C, yes, G, A, T, A, C, G, A, T. Beautiful. All right. Don't forget 5 prime. What would this be? 3 prime, 5 prime. Because it's very important, or else you may get confused. Then the hydrogen bonds are formed in between them. So now a pyrimidine dimer formed here between the both of them. So we found that the multi-enzyme complex recognizes this and initiates the mechanism. The first enzyme that is involved in this mechanism is a nucleus. It's an excision nucleus. Or you can just say nucleus. What it does is that it breaks the phosphodiester bond. So it breaks the phosphodiester bond from not just this region, but from a longer region of the DNA. So let's say from here, breaks here. You see that? That's it. It has broken it. Then, after breaking this phosphodiester bond, we have to remove this, take it off. And we use an enzyme that I know we are much aware of, and we know very well, which is DNA helicase. So you see, so DNA helicase helps to unzip, or let me say peel off. Yeah, I think that's the best word. Peel off. So it simply comes here and peels this thing off. Peeling it off. You start peeling it off gradually, peels it off. All right? Until eventually it gets out of that region. It goes off. Do you see that? That's DNA helicase. It's gone. But we have an empty space here. We can't take this like this. It could be deleterious to the cell. So therefore, we need 2 enzymes. Very important in these mechanisms. There are always 2 enzymes that are involved in the last stage, which is DNA polymerase and DNA ligase. So we have DNA polymerase coming here. Comes here and it synthesizes a sequence T, A, T, G, C, T. Start. So then after that, you see there's still a gap here. The phosphodiester one has to be joined back. So we have DNA ligase coming in to ligate. So it's a ligation reaction. Join this together. Join this together. Finish. So it has been repaired. It's as simple as that. So the next one I'm going to teach you is going to be the repair mechanism for double-stranded breaks. These are single-stranded breaks. One strand. This is one strand, second strand. But there are some breaks that have to involve double-stranded breaks. And if you have heard of CRISPR, the CRISPR mechanism, it has to do with DNA repair because it creates a damage, and during the repair process, that's how the genome is being edited. So, but we'll talk about that later. So let's just stop here for now. Next video you'll see will be on non-homologous end joining.